Hello and welcome dear students. Today we are going to discuss about the topic epistasis and the conversion of genetic variances. The main objectives of today's lecture are to know the basics of epistasis, to know the history of epistasis, to know about epistasis in complex disease traits, to know about epistasis in population genetics, to understand the genetic and molecular causes of epistasis, to discuss genetic variation within natural populations, and to discuss about epistasis and its contribution to genetic variance components. Dear students, let's start with the introduction first. In its original usage, epistasis is referred to the masking or unmasking of the effects of allelic substitution at one locus by the allelic state at a second locus. In modern usage, epistasis refers to any relationship of non-additive interaction between two or more genes in their combined effects on a phenotype. Epistasis is only defined in the context of genetic variation at multiple loci. This variation may be natural or experimental. Epistasis is an important concept in biochemical genetics, population genetics, and quantitative genetics. Although its definition varies somewhat across these fields, the underlying concept is that the effects of allelic substitution at one gene can be dependent on the allelic state of another gene or genes. In biochemical genetics, analysis of epistatic relationships can be used to assign genes to pathways and to define the order of gene action within a pathway. In population genetics, epistasis plays a role in theories of fitness and adaptation. In quantitative genetics, epistasis has taken on a broader meaning that encompasses any non-additive interaction among genes, and it is often identified with the interaction term in analysis of variance. Defining the scale of measurement is important when considering epistasis, as some statistical interaction can be removed by a change of scale. For example, multiplicative effects can be converted to additive effects by taking logarithms. Epistatic interactions can be synergistic, that is greater than additive, or antagonistic, that is less than additive. When two genes interact statistically, it is implied that they must also interact physically, either through direct, that is protein-protein interaction, or indirectly through a network of interacting gene products. Thus, statistical epistasis can provide insights into the genetic architecture underlying complex phenotypes. Dear students, let's know the history of epistasis. Understanding of epistasis has changed considerably through the history of genetics, and so too has the use of the term. The term was first used by William Bateson and his collaborators Florence Durham and Muriel Valde. In early models of natural selection devised in the early 20th century, each gene was considered to make its own characteristic contribution to fitness against an average background of other genes. Because of the way that the science of population genetics was developed, evolutionary geneticists have tended to think of epistasis as the exception. However, in general, the expression of any one allele depends in a complicated way on many other alleles. In classical genetics, if gene A and B are mutated and each mutation by itself produces a unique phenotype, but the two mutations together show the same phenotype as the gene A mutation, then gene A is epistatic and gene B is hypostatic. For example, the gene for total baldness is epistatic to the gene for brown hair. In this sense, epistasis can be contrasted with genetic dominance, which is an interaction between alleles at the same gene locus. As the study of genetics developed and with the advent of molecular biology, epistasis started to be studied in relation to quantitative trait loci and polygenic inheritance. The effects of genes are now commonly quantifiable by assaying the magnitude of a phenotype, that is height, pigmentation or growth rate, or by biochemically assaying protein activity, that is binding or catalysis. Increasingly sophisticated computational and evolutionary biology models aim to describe the effects of epistasis on a genome-wide scale and the consequences of this for evolution. Since identification of epistatic pairs is challenging both computationally and statistically, some studies try to prioritize epistatic pairs. Dear students, let's discuss about the biochemical epistasis. Gene products often act together in pathways and networks. Examples include biosynthetic pathways, signal transduction pathways, and transcriptional regulation networks. Epistasis analysis of mutant alleles provides a means to assign genes to pathways and to determine their order of action. Suppressor and enhancer screens are often used to identify epistatic mutations. A suppressor is a mutation at a second site that causes a reversion to the wild type phenotype and thus masks the mutation at the first site. An enhancer is a second mutation that has a novel phenotype, thus unmasking an effect that cannot be observed in either of the single mutants. Synthetic lethality is a special case in which both the single mutants are viable but the double mutant is not. This can occur when the two loci belong to parallel pathways driving an essential function. Although epistasis analysis explicitly involves only two loci, it can be applied repeatedly to combinations of loci to elucidate the structure of larger pathways and networks. Dear students, let's know about the statistical epistasis. Epistasis in biometrical genetics. In 1954, Cockerham introduced the idea of partitioning the genetic variants from inbred line crosses into additive dominance and epistatic components. Variance components have proven to be useful in predicting the response of population to selective pressure and have been successfully applied in breeding programs. The epistatic variance components reflect an average effect over many genes on the phenotype distribution in a population. Physiological epistasis can contribute to both additive and dominance components of variance, but physiological epistasis must be present in order to generate statistical epistasis. Dear students, let's talk about epistasis in quantitative trait analysis. The availability of polymorphic DNA marker loci distributed throughout the genomes of many organisms enable to track the inheritance of specific loci in line crosses and in pedigrees. Methods for analyzing quantitative trait inheritance using marker data are often based on single gene models that do not allow for epistasis. In the presence of large epistatic effects, these methods may fail to detect important loci or may produce misleading results by statistical phenomena known as Simpson's paradox. Epistasis can be detected using the F-test for interaction in a two-way analysis of variance. However, this test can require large sample sizes to achieve reasonable power. In addition, corrections required to avoid potential false results when searching through all locus pairs further restrict to the power. These difficulties may explain the paucity of reports of epistasis in the quantitative traits literature. This measured genotype approach can be used to make specific predictions about the phenotype of individuals based on their genotype, whereas biometrical analysis can only make statements about population averages. Dear students, let's discuss about epistasis in complex disease traits. Many common diseases show familial aggregation but do not follow simple Mendelian patterns of inheritance. Evidence for epistasis has been reported for traits of medical importance, including cancer, hypertension, kidney diseases, epilepsy, and alcoholism. In addition, there are many genetic modifiers of disease phenotypes that alter the severity of a trait depending on the genetic background in which they occur. Background effects are an example of epistasis. Dear students, epidemiological studies of a common birth defect, cleft lip and palate in human populations have suggested that a single major gene with incomplete penetrance may be responsible for this condition. In a mouse model, the condition appears to be determined by an epistatic interaction between two loci, cleft 1 and cleft 2. When the cleft 1 genotype is small aa, the cleft 2 heterozygote shows a mild form of the condition, and the CLF, that is cleft 2 bb homozygotes, show a more severe form. It is conjectured that these two genes have partially overlapping functions and that the recessive alleles are loss of function. In this example, a model of epistatic interaction provides a testable prediction about the molecular mechanism. Epistasis can be particularly difficult to unravel in outbred human populations, but is often more amenable to analysis in the context of a model organism. Construction of special inbred lines, that is congenic or nearly isogenic lines, is of further use in the analysis of epistasis by reducing the complexity of the genetic background. By its very nature, epistasis is a property of whole genotypes. Epistatic effects are therefore most clearly revealed in the eccentricities of particular individuals. It is perhaps therefore not surprising that some of the best examples of epistasis are emerging from an area in which the focus on the individual reigns above all else is the human health. Dear students, let's discuss about epistasis in population genetics. The quantitative theory of population genetics, as introduced by Fisher in 1918, is based on models of additive genetics in which epistatic effects are represented as a noise term. However, epistasis is known to play a key role in a number of evolutionary processes. Epistasis in traits related to fitness of an individual can lead to the existence of multiple fitness peaks and multiple stable equilibria for gene frequencies in a population. This idea is central to Wright's shifting balance theory. Wright proposed that population subdivision can lead to the evolution of co-adapted gene complexes. Incompatibilities among sets of genes can lead to genetic isolation and speciation. It is interesting to note that epistasis in quantitative traits has been observed more often in crosses between widely diverged strains than in crosses between closely related strains. Epistasis can be beneficial if epistasis is present in a population that has been reduced to a very small size. Inbreeding leads to an increase in additive variance. Thus, hidden variation is exposed to selection and rapid adaptation can occur following a bottleneck. In asexually reproducing populations, the gradual accumulation of deleterious mutations, an effect known as Miller-Rachet, can slow significantly by the presence of epistasis. Finally, the theoretical advantage of sexual reproduction requires that deleterious mutations should occur frequently and that their effect should be synergistic. Dear students, let's understand the genetic and molecular causes of epistasis. First, the additivity. This can be the case when multiple genes act in parallel to achieve the same effect. For example, when an organism is in need of phosphorus, multiple enzymes that break down different phosphorylated components from the environment may act additively to increase the amount of phosphorus available to the organism. However, there inevitably comes a point where phosphorus is no longer the limiting factor for growth and reproduction, and so further improvements in phosphorus metabolism have smaller or no effects, that is negative epistasis. Some sets of mutations within genes have also been specifically found to be additive. It is now considered that strict additivity is the exception rather than the rule, since most genes interact with hundreds or thousands of other genes. Now, let us now discuss epistasis between genes. Epistasis within the genomes of organisms occurs due to interaction between the genes within the genome. This interaction may be direct if the genes encode proteins that, for example, are separate components of a multi-component protein such as the ribosome, inhibit each other's activity, or if the protein encoded by one gene modifies the other, such as by phosphorylation. Alternatively, the interaction may be indirect, where the genes encode components of a metabolic pathway or network, developmental pathways, signaling pathways, or transcription factor network. For example, the gene encoding the enzyme that synthesizes penicillin is of no use to a fungus without the enzymes that synthesize the necessary precursors in the metabolic pathway. Dear students, let's now discuss the epistasis within genes. Just as mutations in two separate genes can be non-additive if those genes interact, mutations in two codons within a gene can be non-additive. In genetics, this is sometimes called intragenic suppression, when one deleterious mutation can be compensated for by a second mutation within that gene. Analysis of bacteriophage T4 mutants that were altered in the R2b cistron gene revealed that certain pairwise combinations of mutations could mutually suppress each other, that is, the double mutant had a more nearly wild-type phenotype than either mutant alone. The linear map order of the mutants was established using genetic recombination data. From these sources of information, the triplet nature of the genetic code was logically deduced for the first time in 1961, and other key features of the code were also inferred. Also, intragenic suppression can occur when the amino acids within a protein interact. Due to the complexity of protein folding and activity, additive mutations are rare. Proteins are held in their tertiary structure by a distributed internal network of cooperative interactions. Epistatic interactions occur whenever one mutation alters the local environment of another residue, either by directly contacting it or by inducing change in the protein structure. For example, in a disulfide bridge, a single cysteine has no effect on protein stability until a second is present at the correct location, at which point the two cysteine form a chemical bond which enhances the stability of the protein. This would be observed as positive epistasis, where the double cysteine variant had a much higher stability than either of the single cysteine variants. Conversely, when deleterious mutations are introduced, proteins often exhibit mutational robustness, whereby stabilizing interactions are destroyed the protein still functions until it reaches some stability threshold. At this point, further destabilizing mutations have large detrimental effects as the protein can no longer fold. This leads to negative epistasis, whereby mutations that have little effect alone have a large deleterious effect together. In enzymes, the protein structure orients a few key amino acids into precise geometries to form an active site to perform chemistry. Since these active site networks frequently require the cooperation of multiple components, mutating any one of these components massively compromises activity, and so mutating a second component has a relatively minor effect on the already inactivated enzyme. For example, removing any member of the catalytic triad of many enzymes will reduce activity to levels low enough that an organism is no longer viable. Dear students, let's understand heterozygotic epistasis. Diploid organisms contain two copies of each gene. If these are different, that is heterozygous or heteroallelic, the two different copies of the allele may interact with each other to cause epistasis. This is sometimes called allelic complementation or interallelic complementation. It may be caused by several mechanisms, for example, transvection, where an enhancer from one allele acts in trans to activate transcription from the promoter of the second allele. Alternatively, transsplicing of two non-functional RNA molecules may produce a single functional RNA. Similarly, at the protein level, proteins that function as dimers may form a heterodimer composed of one protein from each alternate gene and may display different properties to the homodimer of one or both variants. Two bacteriophage, that is T4 mutants, defective at different locations in the same gene can undergo allelic complementation during a mixed infection. That is, each mutant alone upon infection cannot produce viable progeny, but upon mixed infection with two complementing mutants, viable phage are formed. Intragenic complementation was demonstrated for several genes that encode structural proteins of the bacteriophage, indicating that such proteins function as dimers or even higher order multimers. Dear students, let's discuss about the genetic variation within natural populations. From mutational studies, we know that epistasis in the classical sense is ubiquitous because genes interact in hierarchical systems to generate biological function. For quantitative genetics and the genetics of complex traits, however, it is not the total scaffold or biological function that matters, but the residual variation segregating within a natural populations that determines differences among individuals. Traditionally, quantitative genetics has focused on aggregate measures such as genetic variance and heritability to estimate genetic effects. There seems to be little evidence that epistatic variance plays an important role in most populations, although epistasis at individual loci can make significant contributions to additive variance. Dear students, let's know about epistasis and its contribution to genetic variance components. Epistasis is the deviation of two loci genotypic values from the sum of the contributing single locus genotypic values. The contributions of epistasis to the additive dominance and interaction genetic variants are specified. Epistasis can make substantial contributions to each of these variance components. A belief in the importance of genic interaction lies at the core of Wright's ideas concerning the genetic basis of evolution and plays a central role in founder effect models of speciation, that is, the creation of species. Furthermore, theoretical models indicate that with epistasis, population bottlenecks and subdivision expose hidden additive genetic variants to selection, allowing rapid adaptation to new environments. The quantitative effects of epistasis have been difficult to discern by traditional techniques because they are difficult to estimate. Epistatic terms contribute relatively little to the covariance among relatives except when clones are available. The contribution of epistasis to genetic variance components and hence to evolutionary processes remains obscure. To improve the understanding of the role of epistasis, it is necessary to differentiate between physiological and statistical genetic definitions of the phenomena. In physiological genetics, epistasis occurs when the phenotype differences among individuals with various genotypes at one locus depends on their genotypes at other locus. In statistical genetics, the epistatic or interaction deviation is the deviation of multi-locus genotypic values from the additive combination of their single locus components. There are two important distinctions inherent in these definitions. For statistical epistasis is a population phenomena depending on allele frequencies. Present in a specific population, whereas physiological epistasis is a genotypic phenomena independent of allele frequencies at the loci in question. Earlier models of the effects of epistasis have dealt mainly with its statistical effects. Second, epistatic interaction deviations are by definition associated only with the interaction genetic variance component, where physiological epistasis can also contribute to additive and dominance genetic variance components. Dear students, let's now conclude the lecture. Typically, epistasis assists in describing various phenomena such as functional interaction between genes, statistical deviation from additive gene action, and genetic outcome of mutations that act in the same genetic pathway. One of the conventional applications of the analysis as a result of epistasis is ordering genes in a developmental and metabolic pathway. Within the advent of high throughput genetic screens, such approaches of late have turned out to become more systematic. Epistasis can be a roadblock to deduce the genetic basis of complex characteristics in a natural population. Epistasis in human genetic diseases is common, though there are some examples wherein the serviceable footing of a specific interaction has been illustrated. Epistasis emerges as a natural repercussion of the process of evolution, as all resultant changes due to evolution are based on genetic alterations that previously taken place. Dear students, it was all about today's lecture regarding epistasis and the conversion of genetic variances. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.